don't need my computer. You have it there?
Yes, sir.
Wash my hands. I'll be right back. Good morning. How are you, sir?
Good to see you. Thank you for doing this.
Oh, I'm honored. I'm honored. Do you have to sit through all these things?
I get this up too. Yeah. Thanks.
It couldn't be better. You know, just go to work, have a good time, come home, the same thing day after day.
Well,
Too many fun things to work on.
yeah, that's good. Excellent. Well, we're getting rolling here, so I've probably got 25 minutes.
No worries. I'm not worried about finishing lectures anyway. Get too tired on how many slides they have.
So it's a little challenge. We have some people who join us from off-site. So we like to not mute them if we can help it, because then they can ask questions easier. But if they make too much noise, I will come with them, just click the button on the screen. But if you have a question and answer stuff going on, we have these microphones around the room that work fairly well, but they're not perfect. So if you can repeat questions to make sure everybody is hearing this and thing, that would be good. And we can actually even ask if any of the off-site people have any questions. And usually there's silence, and that doesn't do anything.
That's what they're doing their emails.
That's right. They've written a great audio rumor.
I tell people I'll have a discussion in the lecture.
Very good. Okay, let's be careful.
No worries. Happiest man you know.
as we discussed, we are talking about these later stage clinical studies.
Yeah.
We're talking about phase 1 and 2, but I'll just say something about that. But
I can ask an introduction. I have one slide on it, put it in context.
very good. First, we have some couple of pre-docs, a bunch of postdocs, PhD scientists. They
Just have one.
They apply, and they have to have a research project that's either in development or actually that they're doing. That's the support of the involvement to control their time. No, really, they're not in their division director, department chair, because they one day where they go to an IRB session and comes from 9 o'clock till noon. Otherwise, they're here from 9 to 10 after Wednesday all semester, so they have to have commitment to that time. And that's pretty much once had to defer people to the next year because they didn't want to do it.
It's a great problem.
Yeah, it doesn't happen often, but somewhere between 20 and 35 people is ideal.
Now, where are the off-site people?
So some of them are people who are on campus, but just come over here. We have people in the school of nursing, people in recitations of our school of business. I don't think we have anybody from businesses here, but and then we've had people from as partner, she's just part of the work. One person from Mississippi did it once. We've had some occasional people from Arlington. So it's a
Sounds cool.
sort of diversity. Okay. Okay, we'll go ahead and get started. This starts the academic component of the program. It's a great pleasure to have George Howard here to speak today. So this first module is about the general structure of clinical trials. And normally people talk about clinical trials starting with phase 1 and phase 2 clinical trials and then go on to phase 3 and phase 4. We had a scheduling issue this year and realized that really there's no reason that we have to do it in that order. And so Dr. Howard is going to talk about phase 3 and later stage clinical trials. Next week we'll have Mansoor Saleh, who will be talking about phase 1 and phase 2 clinical trials. So I don't think you'll find it confusing in any way. Dr. Howard is one of the giants on the UAB campus. Probably everybody's familiar with him in general, but it's also, I think it's always nice for people to know where people come from. So he did his graduate studies, first getting a master's degree, then interested in an MBA, and Master of Science in Public Health, and then a Doctor of Philosophy in Biostatistics at the University of North Carolina in Chapel Hill. I guess the business degree was at UNC Greensboro, and then he started his faculty career at Duke briefly, and then was at Bowman Gray University in Winston-Salem for most of the development of his initial career, and ultimately in 1999 came to UAB as department chair for this Department of Biostatistics, and I think served there for 8 years or something like that, 11 years. Actually, that's how long I served as department chair. That's the amount of time it takes to accumulate so many problems that you can't solve that you need somebody else to move in. But anyway, he was really transformative to the community here. One of the things that I think is most impressive is that he's been a real national and international leader in very large clinical studies, and he may talk about some of those as part of the discussion today. But the one that's still active after a long time is the REGARD study. He's focused on stroke in a substantial number of his studies, but he'll tell you, I'm sure, a little bit about that. One of the things that's the most impressive to me is the extent to which he has recognized the phenomenal value of large studies to the entire research community and has tried to make sure that databases that get generated in his own studies get made available to other individuals to work with. And so he'll, I'm sure, make some comments about that in his comments today. And we'll, as usual, we'll try to take a break towards the middle, until 9 or 9, and don't hesitate to ask questions as we go along. He's very eager to help.
No, I was going to start off by saying I'd rather have a chat than a lecture. I really, really want to invite you to interrupt me at any time if things aren't making sense. This is sort of a weird lecture or a weird chat, if it's going to be a chat, because basically I teach a one-semester course in clinical trials, and then Gary Cutter teaches a two-semester course that follows that one, so 3 semesters of clinical trials, all of which you're going to get in 1 hour or 2 hours. And so this is sort of, I was going to say, the subtitle is wandering through about 1,000 different topics that at some time will seem a little bit unconnected. But so basically, I think it's important, very, very important, to put clinical trials in the right context. And so basically the way research happens is it starts with ecologic studies. Just basically either clinical observations, you see some patient who has some condition and lo and behold he has some gene or some risk factor, and you say, I wonder if that's it. And you move from there to observational epidemiology, so a field that I really love. Within observational epidemiology, basically observational epidemiology is exactly what it says. You're observing, you aren't manipulating, but you're just observing. There's also a hierarchy of evidence within observational epidemiology that moves from a case control study, which is considered the weakest of the observational epidemiology studies, to cross-sectional design, and then the gold standard of observational epidemiology is a prospective cohort study. I'm sure you'll get lectures on each of these as you move forward also. And then finally, you get the randomized clinical trial. And then I'll guarantee you, if you haven't heard it now, you'll hear it 1,000 times. This is the gold standard. This is the gold standard. And I think if you don't get anything outside of this lecture, what I'd really like you to get is why this is the gold standard. And so basically, the teaching example of my tenure on faculty has been a hormone replacement therapy. There were just tons and tons of very, very well done observational epi studies, all of which showed hormone replacement therapy protects women from heart disease. In fact, I stood it before the Women's Health Initiative started. I'm embarrassed to say that I stood arm in arm with a wonderful woman, Trudy Bush, at the American Heart Association epidemiology meetings and said, we need to block the Women's Health Initiative. We all know the answer to this. Hormone replacement completely protects women from heart disease. This is an unethical study, and we need to work together to make sure this study never gets done. Well, thank goodness nobody paid attention to me. And the Women's Health Initiative was done. In fact, 2 other studies were done, all of which showed that hormone replacement was not only not beneficial, but it was perhaps harmful. Okay, the joy of science, by the way, is being wrong. I absolutely love being wrong. When you realize how wrong you were, that's when things are really moving forward. So that's something to be celebrated. And so the question is, the entire field accepted these 3 studies. Women's Health Initiatives was big. Hers and I'm blanking on the 3rd study, were little teeny studies, not very big studies. And so how can this occur? How can observational epidemiology be so wrong? And why do we believe 3 studies, 3 randomized clinical trials that are much, much smaller than these very, 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 very well done observational epi studies? And the answer to that is largely confounding. Observational epi studies are subject to confounding. If you don't get, and this is the slide I want you to take home. This is the important slide. So what I'm interested in, is this a pointer?
Actually, you got to use the arrow on the keypad.
Okay, don't worry about it. So basically, I'm going to use this pointer. So basically, so I have a risk factor and an outcome, estrogen and heart disease risk. And what I'm interested in is this relationship. Okay? Unfortunately, there are other things in the mix. And one of the important things in the mix that I think is playing an important role in this is socioeconomic status. Because it turns out, at least in America, rich white women take hormone replacement or took hormone replacement. And rich white women also tend not to have heart disease. Okay? And so basically what happens is you can think you see an association between risk factors and outcomes, not because it's really there, but because of this nasty confounder. This is one of the key parts. key concept here. A confounder is anything that is associated with both the exposure and the outcome. Some people define it slightly differently, but this is the right definition. So basically anything that is associated with both the exposure and the outcome. Something cannot be a confounder unless it's associated with both the exposure and the outcome. Okay? So in this case, rich white women are more likely to use hormone replacement. Rich white women are less likely to have heart disease. So it could have looked like hormone replacement prevents heart disease. Not because it did, but because this pathway through rich white women. Okay? So how does randomization fix the confounder? And in randomization, what you're doing is assigning the exposure, in this case estrogen, to the two groups at random. So basically, there can be no association between the risk factor and the confounder. The exposure and the confounder. You've broken that leg of the table out of the way. It doesn't matter that confounding is still related to the outcome. It is, but it cannot be a confounder unless it's associated with both the exposure and the outcome, and randomization fixes that. That is the reason. This is the reason that clinical trials are the gold standard. Okay. Now, randomization is wonderful. They're really. It's amazing what it does. I mean, it really is amazing. It's wonderful stories about randomization. It was really invented by a guy named R.A. Fisher, who, by the way, invented about 3/4 of statistics while he was at it. He was at a dinner, and this woman had a cup of tea brought out to her. And she had it sent back because she said that the cream had been added to the tea instead of the tea added to the cream. And she greatly preferred her tea added to her cream instead of vice versa. And so at the dinner table, their argument came up that this woman was nuts, basically, in different words. And everybody said, you can't do this. And so R.A. Fisher, what he did was have 6 cups of, by the way, there's actually math behind this, but had 6 cups of coffee brought out and presented to her randomly, half of them tea to cream and half of them cream to tea. And she tasted them, and by the way, got it right. And so basically, apparently this woman could see it, but that was really the beginning of 2 concepts in statistics. Really, one was randomization, and the second one is something called a permutation test, but that's not for today. But anyway, but the key to it is not only is high SES equally assigned to the 2 groups, but there's equal exposure for all risk factors. And the key to it is even the ones you don't know about. Okay, randomization not only fixes the risk factors that you know about, it fixes the ones you don't know about. You could find out in 10 years that there's a new risk factor and this is really related to heart disease. That got assigned to the 2 groups at random also, even though you didn't know about it. That's the cool thing about this. So why don't you always do randomized clinical trials? Well, there are lots of reasons. First of all is this idea of the spectrum. These things are expensive and you don't start at the end of the path. You work your way up to the path. The second is that they're really expensive. The trials that we're doing right now in carotid disease are run in the $60 million range. And so you don't, decide, let's do 30 randomized clinical trials for all of these different things. And importantly, this is a slight lie. Randomized clinical trials only address one question at a time. There's only one exposure, generally, in a randomized clinical trial. Get to that later. And some topics can't be addressed by randomized clinical trials. It's amazing to me, I used to live in Winston-Salem, North Carolina, a town that the cigarettes were named for, not the town named after the cigarettes. Okay? But it turns out that the tobacco companies are really smart. And what they say is there's not definitive evidence that cigarettes are related to lung cancer. How many people believe that? I do. The only way that you can definitively prove this is to get children and randomly assign them to smoke and not smoke, and then follow them for their lung cancer. So they are correct. I mean, it's a stupid argument, but it's a correct argument. Okay. So cigarette smoking, there's no randomized clinical trial evidence for seat belts. There's no randomized clinical trial evidence for parachutes leaving airplanes. But we know certain things work because of observational epi studies. Okay, so now this, by the way, is something David was talking about. Next week, you're going to be talking about phase 1, phase 2 studies. And this is just one slide to try to put this in a little bit of context. Is you also don't wander into clinical trials and do these big phase 3 trials first, which is what I'm talking about. The first thing you do is make sure that the drug doesn't kill people. And so generally, phase 1 studies are generally the drug given to a very small number of people who, at least in the cancer world, are the sickest of the sick, to make sure that the drug is tolerable, can be tolerated. Okay? You move from that to phase 2, which is generally, by the way, these terms are very poorly defined. If you get out 3 or 4 textbooks, you'll see different definitions for phase 1, 2, 3, 4 across textbooks. There's not, people don't really agree about the definition. Phase 2 is the early efficacy. The first step in this is dose finding. So you're trying to move from this drug works in a rat to how much does this drug work in a person. And the first question is, I know how much drug to give a rat, but how much drug do I give a person? The hot topic here is something called continual reassessment methods and really spiffy ways to do dose finding in people. You move from that to phase 2B, which is the early efficacy studies. Early efficacy studies. Before I do the big trial, I just want to see if I think this thing works. And so here I do randomize people, but generally I have a surrogate outcome. I'm interested in whether or not this prevents stroke. But for the surrogate outcome, I might be interested in, does it stop the progression of carotid atherosclerosis, which I think is on the pathway to stroke. And so what you're trying to do is you don't go after the main outcome. You go after surrogate outcomes that would give you an indication that this might work. Okay. You move from that to pivotal clinical trials, which is the phase 3 trial, topic of today. And basically the goal here is efficacy with a little bit of safety and a little bit of regulatory going on. One of the important concepts of phase 3 trials, BIOPS is the teaching example of this, is you don't really have enough safety events, we'll talk about this a little bit later, to really get a clear picture of what's going on for the safety. There won't be enough heart attacks in a BIOPS trial to find out if it really works. It was powered for the efficacy outcome, which was pain relief. Okay? So then what happens at the end of the phase 3 trial, you say, oh, this is a great trial, drug, it works to prevent its primary efficacy outcome, but does it work for safety? Are there safety concerns? And so that brings up the phase 4 trials, which are really called post-marketing studies. And basically what you're doing there primarily is looking to see the safety of the drug. You can't really get the safety of the drug completely out of a phase 3 trial. It's not what it was designed to do. That's why you do the post-marketing study. Yes, ma'am.
So how is 2A different than 1? Because I would have always thought dose finding would have been in 1.
Oh, these two? Dose finding is generally you're giving, there's no randomization other than to the dose. And so what a continual reassessment method is really a cool idea. It's exactly what it sounds like. What dose do I give these people? In my world, I live in the world of stroke. The bad outcome in stroke is having a hemorrhage. Okay, I'd like to pick a dose that, through our colleagues in cancer, there's a belief that more of a drug is better than less of a drug. And so what I'd like to do is to push the envelope as far as I can to give as much drug as I can without having serious adverse events. Okay, so dose finding is I want to know how much TPA I can give somebody without increasing the risk of their hemorrhaging.
So in phase 1, you usually give a standard dose.
Oh, phase 1 is 20 people, and I'm just going to give it to them right before they die and see if they're okay. I mean, it's first in humans is what it's also called. Just in humans.
Yeah, so what about, how do you choose what dose to use for phase 1?
Yeah,
Is that like a middle road?
Well, this will be next week too, but frankly, what you do is you say, number one, I tend to do phase 3, but you say, gee, a rat weighs this much and you weigh this much. Let's multiply the dose we give in a rat to your level. So yes, that's why you do the dose finding next. Okay, so basically, believe it or not, this is the outline of my talk. This is going to be, like I said, 1,000 topics, sort of in high speed. I'd like to talk about the hypotheses, the eligibility, subgroup analysis, endpoints, blinding and masking, analytic issues, novel study design, and data and safety monitoring. It's all in an hour and 45 minutes. Okay, so starting off with the ethics of this, the key to randomized clinical trials is equipoise, and equipoise is frequently misunderstood. Any of y'all surgeons in here? Okay. You know, I hope I never am cut on by a surgeon who has any doubt. You know, I don't know if y'all deal with surgeons, but you don't run into surgeons who say, I'm not sure what I think about this. I mean, the surgeons, I've never met a group of people who are more confident they're saving people than surgeons. And by the way, that's good. I want them to feel like that. Okay, I want them to feel like that. It's not an uncertainty in the scientist. It's equipoise is an uncertainty in the community. We can't agree whether this works. There'll be individuals who think it does work and doesn't work in every trial. And what you want to do, by the way, is be careful how you manage those people, because those people can distort a trial. But anyway, it's the general uncertainty in the expert medical community of which is better. Surgeons already know that they are saving people in every surgical trial I've ever been part of. So equipoise, clinical community versus the uncertainty principle. One of the things, I wish I had more time to talk about it. In Europe, they approach things slightly differently with something called the uncertainty principle. In Europe, individual clinicians put people in the trial that they are uncertain how to manage. Okay? So that means that Bill and Susan, two clinicians, Bill might be putting sicker people in the study than Susan, because Bill, you know, thinks that this might be working in a different group of people. Okay. In America, we try to have a protocol and define what the eligibility is, and then we try to work very diligently if somebody is not putting eligible people in the study to modify their behavior. Okay, so in America, what we're doing is equipoise and trying to randomize all eligible people. In Europe, they use this thing called an uncertainty principle, where the individual clinicians play a larger role in who gets in. By the way, big difference between the two studies in Europe and America. Big difference. Okay, and then of course informed consent is key to this. I imagine you'll have whole lectures on informed consent, but basically the things that we have historically done to people have been shocking in terms of getting people in studies that they don't know what they're doing. And so basically the hypothesis in terms of the characteristics, it's writing a hypothesis is sort of a high skill task, and it's really important to do. It needs to be clear, quantifiable, simple, focused, singular, and basically have the who, what, when of the study. This is for CREST that we're doing, which is contrasting stenting versus endarterectomy to prevent stroke, myocardial infarction, and death. So we've said exactly what the outcomes are. During a 30-day procedural period, this is the when, and stroke ipsilateral going up to 4 years, and in the WHO is in symptomatic and asymptomatic extracranial carotid atherosclerosis. It's sort of fun to read hypotheses of studies and sort of think about you should be able to read the hypothesis and pretty much know what the study is all about. It should flow fairly evenly. I want to take a minute, I debated not doing this, but I think this is another one of the, by the way, statistics is a very trendy field. You know, things get hot. And so one of the hot things that has really been hot for a little while is the idea of superiority versus equivalence trials. Okay? Statistics does certain things very well and it does other things very poorly. A superiority trial has been designed to address the question, are these two treatments different? You know, these two treatments different. And so basically you do that by the way, all of statistical testing is whatever you want to prove is true, you assume is false. and collect data and see if that aligns with your assumption that it's false. And so in this case, you assume the two treatments are the same because I'm trying to prove that they're different, and I collect data to see if they look different. If they really do look the same. I've said that backwards. I'm sorry. Let me fix what I said. I collect data to see if they really do look the same. Okay. It turns out very frequently in statistics or in medicine, you're not interested in proving if things are different, but proving if they're the same. And the example of that I've run into is carotid stenting. Okay? All things being equal, many people would rather have a carotid stent than an endarterectomy. I mean, it's less invasive. You don't have to have your neck slit open. A lot of other benefits. And so if stenting is as good as surgery, you probably should do stenting. We're doing a trial right now that is endarterectomy versus medical management, okay, with really aggressive medical management. All things being equal, I would like to avoid surgery, okay? So basically, if the medical management is as good as the surgery, I'll vote medical management for me. And so I don't need to prove that medical management is better. It'd be nice if I did. But if it's just as good, then I'll go that way. So there's a whole field of trials called equivalence trials. And suppose that a new treatment has fewer side effects or lower costs, then showing that the new treatment is as good is really a win. All I have to do is say medical management is as good as surgery, and I'll do the medical management. Yes, sir.
Is there an ethic behind that? Reason being, so let's say now medical management is better. There are fewer surgeons doing endarterectomy, which means you are getting trained in that, which means it sort of is a self-fulfilled prophecy that then if you need it, you don't have as good a surgeon, so you're actually harming the medical community, perhaps.
Well, that's a good point, and I mean, I think, if you talk about this, okay, here's the radical socialist statistician. So I think we do surgery wrong anyway. I think what we should do is to have something like 10 endarterectomy centers in the nation. If you want to have it done here, you go to Atlanta and have your endarterectomy done, and we ought to have people there who wake up in the morning and do that one surgery all day long, and then go to bed and wake up in the morning and do that surgery all. But the idea that somebody could do 3 endarterectomies in a year, I think is a dangerous concept. I mean, I know I'm talking, about reimbursement and other things that get complicated, but if we really wanted to care about our patients, we would go down that pathway. It would be a pretty big disruption in medical delivery of medical care, but I'd love to hear the argument against it and why we should let somebody do 5 endarterectomies in a year. Yeah.
In an equivalence trial, if one turns out to be better than the other, which is not what you were expecting, can you claim superiority in that?
Yes, you can. Yeah, I wish I'd have brought copies of it, but there are 2 important papers. Write this down and pull them up. There are things called consort guidelines, CONSORT. I should have had that as a handout. I'll do that next year. But there's a consort guideline for trials and a consort guideline for non-inferiority trials. And basically that's the rule book. I mean, you follow those. But yes, you can. Good question, by the way. Okay. Now, the trouble with traditional trial design is there are 2 reasons why a trial can be negative. A superiority trial, the first one. The first one is that the 2 treatments are really the same. And the second is I didn't do a big enough study to see that they're different. Okay, and so basically one of the things that statistics does poorly, again, is this idea of proving that things are the same, because really you can't tell which one of these are true. You never accept the null hypothesis in statistics. You only fail to reject it. I fail to reject the null hypothesis. And so if I do a superiority trial and they're not different, I don't say that they're the same. I say I fail to reject that they're different. Okay, it's a double negative. It's a horrible double negative. The cool thing is that if I'm doing an equivalence trial or non-inferiority, my null hypothesis, as you will see, is that the 2 treatments are different, and the alternative is they're the same. So what I do there is reject the null hypothesis that they're different and accept the alternative hypothesis that they're the same. Okay, and that implies that traditional studies cannot, the superiority studies cannot be used to show that a new product is as good as the old. So basically the answer is to change the form of a hypothesis. If you're doing a traditional trial, this was invented by a guy named Blackwell. What he did was flip everything around and sort of backwards thinking. The first time you see it, you have to sort of ponder it a little bit. But what he did is say the null hypothesis is the new treatment is worse than the old. And the alternative hypothesis is it's as good or better. And the key phrase there is the as good part. Okay? And so now if we reject the null hypothesis, we definitively conclude that the new treatment is as good or better than the old, and I get what I want. It's the as good part is what I'm really after. It's simple in concept. There are lots of different names for it. Some people call it non-inferiority, others call it equivalency, people call it Blackwell's of trials. Pretty much the same thing. Pretty much the same thing.
But this does not take care, changing basically null to alternative to null does not take care of the inherent problems with your sample or population that you're running, the power issue and all those kind of stuff.
Yeah, but what we're going to do is design, I'll show you how we set this up, but what you do is design it. What happens is we end up having to define the same in a little bit of a counter. The same sounds like a simple concept. It turns out not to be. But so we're going to define the same and then get a large enough sample to prove that it's the same. Okay. So regardless of the form of the hypothesis, the study provides an estimated difference between the two treatments. All the information that is in a study is the estimated difference in the 95% confidence limits around that difference. Okay. That's all there is. And so basically I can link the hypothesis testing, and if something is inside of the 95% confidence limits, I will always fail to reject. If something is outside, if the alternative hypothesis is outside of the 95% confidence limits, I will always reject. Okay, so I can either do this through testing or looking at confidence bounds. Looking at confidence bounds makes this easier to explain. Okay, so basically this is the simple clinical trial, 2 treatments. I have an established treatment and a new treatment. I have a dichotomous outcome in my example. Percent of the people events in the established treatment is pi O and pi n is the proportion that are having events in the new treatment. And in general, which treatment is best? The way that you do this is the null hypothesis is there's no difference between the 2 treatments. Those 2 proportions are the same. Okay. The alternative hypothesis is they're different, which just says they're not the same in either direction. Okay. And when I test that, if you look at it, what will happen is this is no difference between the treatments. New treatment is better. Old treatment is better. And so basically step 1 is I calculate the difference. And so there it is. That's my example of the difference between the 2 trials. I can then calculate 95% confidence limits around that point like that. And if I do a traditional trial, if 0 is not in the 95% confidence limits, then it's significantly different. Okay? So if I do a traditional trial, 3 things can happen. Okay? Thing number 1 is I can have the new treatment better. Thing 2 is I can have no difference. Or thing 3 is I can have the old treatment better. And it's just simply where those 95% confidence bounds are falling. This is easy. This is easy. Okay? So the advantages and disadvantages of a traditional study. The advantage of a traditional study is I can always definitively state that one treatment is better than the other. I can really make a strong statement there. The disadvantage is if there's not a difference, I can't state that the 2 treatments are the same. Again, null hypothesis never accepted. Okay? And you end up making a statement that is really sort of a kissing your sister kind of statement, something that makes everybody unhappy. There's not sufficient evidence in these data to establish that a difference exists. Okay? That's not a real strong statement. Okay? The manufacturer, of course, sometimes wants to say things are at least as good, and that's why you do equivalency analysis. And here, again, it's to say that things are as good. We've already done this. So what you do is you make the null hypothesis be that the new treatment is worse, has a higher event rate than the old treatment, plus a little fudge factor, or cushion, a delta. Okay? The alternative hypothesis is the new treatment is as good or better than the old treatment plus a cushion, which is going the other way. That delta is why I'm not a big fan of equivalency trials. So this is, the FDA is, by the way. Okay? But at the end of the day, you have to end up defending your delta. Because really what you're saying is these two treatments are the same. And by the way, when I say the same, I really mean within 2%. And so if somebody else comes up and says, what do you mean 2%? We're talking about killing people. You can't kill 2% more people. The same is within 1%. And so you end up having to defend your delta in these equivalency trials. Okay? The definition is as good really means that it's within the cushion. The FDA, for some reasons, good arbitrary numbers tends to like 2%. I mean, they at least in my world, that deltas are generally around 2%.
So that 2% was just sort of arbitrarily.
Right. By the way, do you know where 0.05 came from in statistical tests? Same R.A. Fisher do. When it was all being worked out, Fisher was literally at a conference, and somebody said, this is really a great idea, this statistical testing. Professor Fisher, how much evidence does it take for you to reject the null hypothesis? And his quote, this is supposed to be a quote, is, gee, I don't know, 1 in 20 usually does it for me. Okay, and from that we got 0.05. Okay, and so things end up getting chiseled in stone, which by the way is goofy. I mean, so let's say that I'm interested in whether orange juice prevents colon polyps. And I have something that says orange juice is better if P equals 0.1. Would you recommend taking orange juice? I would. I sort of like it. You know, it tastes good. No real risk other than the sugar. I guess you could argue the sugar's not good for you. But let's say that what you're doing is splitting children's chest open to do some procedure. Is a 1 in 20 chance enough to really say I recommend that every child in America should have their chest split open? You know, 0.05 is a goofy, really goofy number. Okay, so what you're doing here is, this is the same idea, is the idea of looking at confidence bounds. What you're doing in equivalency testing is I still calculate the difference between the two treatments. There that is again. And now I calculate the one-sided 95% confidence bounds. And so what I'm saying is I'm 95% sure that it's at that point to infinity. Okay, that's my one-sided 95% confidence bounds. And if that confidence bounds includes delta, I say it's not the same. If it doesn't include delta, I say it is the same. Okay? I might have said that backwards. Let me make sure I said that right. I'm scared now. If it does not include delta, as this, you would say that's evidence that they're the same. If it does include delta, you cannot say they're the same. So basically, there are two outcomes in equivalency testing. The new treatment is as good or better than the old, and the new treatment is not as good or better than the old.
Excuse me, is this something like non-inferiority studies?
This is a non-inferiority study. The non-inferiority, Blackwelder, and equivalence are all the same. Yes?
I apologize. So the same that we said for the traditional approach, you can't say that it's worse. You can just say you fail to reject null hypothesis.
Yes. Okay. Yeah, you never accept your null.
And in this one, the 95% confidence interval always crosses 0.
Well, if you think about it, let's say that they really are the same. Let's say they're spot on perfectly the same. Then what you would anticipate is my estimate, this orange bar would be right on top of 0. And so that's why you need the delta, the fudge factor, is the uncertainty that it's on the other side. That's why you need that fudge factor. Okay? So the advantages of equivalence is I get to say it says good. The statement largely meets the needs of the manufacturers, and my little people it makes sense. And then that brings up this idea of one-sided versus two-sided hypotheses. And so basically, let's assume there's a new treatment and an old treatment, and the outcome is the proportion of events. A two-sided hypothesis is what we're used to thinking about, where basically the null hypothesis is the two proportions are the same. The alternative hypothesis is they're different. That means one is bigger than the other, but I'm not going to say which one. A one-sided hypothesis is you assume the new treatment is equal to or worse than the old, and you test if it's better. And so basically, it would seem like a one-sided hypothesis would really be a good idea. Let's go back to my example of surgery versus medicine. You know, why do I need to prove that medicine is better? Okay, I only need to prove if surgery is better. It would seem on the surface. Okay, and so assume that the new treatment would not be used unless it's clearly better than the old. Assume that surgery won't be used unless it's clearly better than medicine. Okay, for example, you know, new treatment is surgery. This is the study that this was done in. By the way, right when we did this, 2020, which is it's no longer on the air, but it's a news show like 60 Minutes used to be. Had a 1-hour show 2 weeks before we started randomization that talked about the number of people who were being killed in endarterectomy with this needless surgery. It's like the ultimate bad timing for this to happen. You know, surely surgery wouldn't be used unless it's proven to be better. So it would seem that a one-sided hypothesis is logical. Okay. So why would you use a one-sided hypothesis? If you do a one-sided hypothesis, it restricts the alternative space and makes it easier to find the difference. In essence, you're testing at the 0.1 instead of the 0.05. Has that effect? And so you can shrink your sample size. You know, if what I'm doing is a one-sided hypothesis, a two-sided hypothesis, trying to detect the differences that are in this statement, I need 1,000 people. If I do a one-sided hypothesis, I only need 800 people. It's cool. Okay. If you're not interested in increases from the risk of the surgery, this is a 21% reduction in the number of patients who have to be exposed. There's also the ethics. Okay. For this. When should you consider using a one-sided hypothesis? Never. Never, never, never. Never, never, never. Okay? You know, why not? To begin with, you're providing valuable proof. If medicine is better, if I really want to stop surgeons from doing endarterectomy, if I say the two things are about the same, these surgeons, again, are very competent people. They're going to continue to do operations. Good for them, by the way. Not a criticism, just an observation. If I say medicine is better, I can stop them from doing the surgery. Okay? So you're providing valuable proof. A one-sided hypothesis was not used. You can't definitively prove surgery was inferior. In this case, it's difficult to discourage the procedure unless it's proven harmful. And also, it sort of smells bad. A one-sided hypothesis, you'll have a hard time getting it accepted in any journal because everybody will think you're cheating. And so
So even for the non-inferiority studies, even though you
That's
only have a one-sided 95% confidence interval, you still do a two-sided?
No, that is the exception. Okay. That is the exception. Okay. The one thing that's really important is if you are going to do a one-sided hypothesis, whatever you do, write what is called a design paper, you know, describe your study, and have it published before you ever see a patient, so that you're on record as saying you're going to do this. Okay, so basically we finished the hypothesis part and we have killed a lot of time. Okay, eligibility. Who do you want in your study? Who do you want to have in your study? Okay, so basically what you have here is competing demands. You would like to have homogeneity in your study. Really, you'd like to move your clinical trial more like it's a lab rat study. And a divergent group of patients can really make finding differences in a clinical trial very difficult. So what I'd like to do is to have the eligibility criteria select people who are as homogeneous as possible. Okay? The restriction in the population reduces what the statistical noise and will make it easier to find a difference. This is why, by the way, a lot of studies exclude the very, very old and the very, very young from trials. If you talk about, in my world, stroke, people who have stroke at age 20, could be sort of drug related instead of atherosclerosis related. It could be a different mechanism altogether. And so basically restrict the population to a homogeneous group. That's what I want to do. While I'm doing that, what I'd like to do is ensure the generalizability of my study. Okay? And I'd like to, at the end of the study, I'd like to apply these findings to a broad group of people. Okay? You know, if I exclude young people from my study, I can't say this study applies to young people. Okay? It's questionable to generalize the findings to people who are excluded, and therefore my second goal is to have my study be as broad and as inclusive as possible. Okay? Obviously, these things are in direct contradiction. I mean, these are pointing that way. Okay? And so what's the correct answer? And the answer is there is no correct answer. You have to fight your way through this and think through what will happen at the end of my study and who do I want to generalize to and who do I want to have my findings apply to and who do I not want to have it apply to. And it's so fascinating because in my world of revascularization, the studies that were done during the 80s and 90s excluded people at age 70. Basically, over half of the procedures that are done now are done in people over age 70. And so the second wave of studies are finishing up, but there was no clinical, phase, class 1A evidence that these things were good for half the people receiving the surgery. Not a good situation. Clearly a balancing act. You have to think of how weird the weird patients are. I beg your pardon. Will the results be accepted for all the subgroups? Is there really one disease? Stroke as atherosclerotic versus stroke as cocaine. Those the same drug. Will subgroups analysis be required? You have to just use good judgment, and it's difficult to depend on subgroup analyses, but we're going to talk about subgroup analyses shortly. Okay, speaking of.
So, just in general, if you create a study that's extremely homogeneous, have there been studies to see if that reduces the likelihood that the outcomes will be safe in the end? Or it seems like
a study would be completely applicable to the people included in the study. So if I make it homogeneous, what I'm doing is reducing noise. So I can do this in a smaller group of people, and I'd have an absolutely appropriate answer to people with those eligibility criteria. But if I don't have those eligibility criteria, it's interesting. One of the classic studies of all time was something called Mr. Fit. Anybody heard of the Mr. Fit study? This is a great study. So what determines the power of a trial is not the number of people in the trial, but the number of people who are having events, the number of people who have heart attack and stroke. A study of 10,000 people where 50 people have a heart attack has the same power as a study of 100 people where 50 people have a heart attack. Okay, it's not the number of people, but the number of events. So when you're planning these randomized trials, one of the things you're thinking about is, how can I enrich my sample? How can I get people who have more events? Okay? Well, one way to get people who have more heart attacks is only study men. Men have more heart attacks than women. And so the whole idea of Mr. Fit, unfortunate name of the study also, was they studied a gazillion men to find out if primary prevention could prevent heart attacks. And then what they said is, it's okay, women are just like men. We can generalize this finding from men to women. That didn't go down very well in a lot of groups. That could have been a very controversial trial with really poor outcomes in terms of the interpretation. It's been only done one time like that I know of, but that was a good, I mean, it was done with good intent. It was done to expose as few people as possible to these procedures and do an effective study. It's just you might want to think women and men are different and be able to generalize to women. But if you do that, what you'll have to do is a subgroup analysis. You would like to look at men and women separately in your trial. The catch-22 here is, by the way, I'm not arguing against subgroup analysis. Absolutely logical, necessary. Are there groups within the study where the treatment was differentially efficacious? Does this have the same benefit in men and women? Were there groups of the study where the treatment was ineffective? These are natural questions, but you really have to plan them. You really have to plan them very, very carefully. And so basically what we're talking about was, some people at this point say, I thought you said confounding is not an issue in a trial. This is actually not confounding. This is effect modification or interaction. So you can have interaction in a trial, but not confounding. Okay.
Question.
Yes.
Are you talking about post hoc analysis, or are you saying a priori you're going to establish that you're going to do a subgroup analysis?
Great question. Okay. And I tell you, can I table it just for a second though? Okay, so basically examples are, let's say you're interested in diabetes and smoking to see if they're related to stroke and extracranial atherosclerosis. So I'm going to perform an analysis only in the medically managed people at a study called ACAS that I was part of. And this was a randomized trial of surgical management of carotid stenosis. And there were 800 people managed medically. There were 21% of the people were diabetic. And this is an example of a context of an analysis in one arm of an RCT. The same thing is present if you're doing subgroup analysis of an entire trial. So let's say that, the reason why I was interested in this is, if y'all don't know Elizabeth Barrett Connor, everybody needs to meet Elizabeth Barrett Connor sometime. She's this woman who knows everything. I mean, she is unbelievably brilliant. And she wrote a paper one time that said, diabetes is bad for you, and cigarette smoking is bad for you. But if you have diabetes and cigarette smoking, you're toast. I mean, there's more than additive. It's more than additive. Okay, and I'm not going to read all of this because I'm running out of time, but that's basically what this says is, this in your handout. Basically, if you have both of these, you're really going to get in deep trouble. It's bad to smoke, it's bad to have diabetes, but if you're diabetic, by God, don't smoke. That's why I'm interested in this. And so what I'm interested in is doing a subgroup analysis to see if there's increased risk of cigarette smoking is greater among diabetics and non-diabetics. Okay, and my hypothesis is the time to stroke will be shorter in those people who have both. And so I've basically done this very standard analysis. This is the overall result. These are real data, by the way. These are real data from ACAS. So this is overall for normal and diabetic, and you can see there's no real big effect of diabetes in ACAS. There's non-significantly different. And there's 43% increase, but it wasn't significant. But when I look at it stratified by status, here it is for my non-smokers, and here it is for my smokers. Okay, not good, not good. In other words, if I've got both of them, my hazard ratio for smoking, if I'm diabetic, is 2.73. Not good. Really for diabetes. So the conclusion, how many people think I can write this up? I do, by the way. Okay. So there was a trend for diabetes to be bad for you, but basically, but it's primarily driven by smokers, where diabetes is 3 times worse for you if you're diabetic than non-diabetic. Real data, by the way, real data. And these were confirmed by previous literature. I've got Elizabeth Barrett Connor's really cool paper. I think she's a brilliant woman. Great paper, great paper. So it confirms my belief that diabetes is important, and it underscores the importance of smoking cessation in diabetics. I'll bet you $1,000 I can get this published. I bet I can get this published in a good journal, Okay? So what really happened in preparation for this lecture? Okay, to begin with, I lied. All of that was a lie. I first did an analysis of hypertension, okay? Overall it was non-significant, and I looked in other subgroups to see if it was significant. In fact, I looked at smoking, but I also looked at sex, race, Ruiz, alcohol intake, height, weight, 1,000 other things. That took me. By the way, nothing really excited me. I did 44 separate statistical tests, and nothing was really that interesting to me. Okay, it took me about 15 minutes to do that. It's really easy to do. Okay. Next, what I did was look at cigarette smoking, and overall it was non-significant. I did the same 44 analyses there, so I'm now up to 88 analyses that I've done. It took me another 15 minutes. Finally, I got into the idea of diabetes. Okay, overall it was non-significant. I found the association in smokers, and then I remembered Elizabeth's paper. Okay, at that point I said, God, Elizabeth Barrett-Conner has written about this. But I didn't do that until after I saw the effect. This is your post hoc idea. I ignored the significant findings here. By the way, you didn't ask about them, so I'm not going to tell you in my paper. It was also diabetes was differentially harmful in ever smokers and people with carotid endarterectomy and people with Ruiz. It was in whites only, people with hypertension, the middle tertile of weight, by the way. I mean, it wasn't important in fat people or skinny people, but the middle tertile, the effect was there. That one didn't excite me, by the way. Okay. The mechanisms, by the way, one of my favorite stories in ACAS was at the end of the study, what we did, again, endarterectomy in medicine, we found that completely unanticipated effect modification with sex. So that it was efficacious in men, but not in women. And my mentor and the guy in my career, Jim Toole, he's from a different generation, and he says things in a funny way. But he said the reason for this is women have delicate arteries. And by this he means they're physically smaller, is what he was trying to say. He used an unfortunate term, delicate arteries. And surgery in delicate arteries is more challenging, and that's why it didn't work in women. And I stood there with him for a minute, and I said, Jim, what would you have said if it had been efficacious in women but not men? He stood there for a minute, and he said, then I would have said, men have more advanced carotid atherosclerosis, and operations in men with more advanced atherosclerosis is risky. Once you see the outcome, I can explain why it happened very easily. So basically, I did more than 100 analyses. It took me about 40 minutes. That confirmed, I only reported things that confirmed my biases and my sense of the literature. I ignored all the other stuff. Was I cheating? If so, how bad did I cheat? And so this is the important figure. If I do one statistical test, I'm right here, and the chance of me falsely reporting something that didn't really happen is 5%. If I do 10 tests, the chance that I find something is about 38%. By the way, I did 100 tests, so I'm way over here. Basically, what you have is a coin that has a 5% chance of landing on heads. And if I flip it one time, I have a 5% chance of getting a head. But if I flip it 10 times, I'm almost certainly going to get a head, which means I'm going to find something going on. So going back to your very good question a minute ago, it is critically important to, I flagrantly cheated. By the way, these results are still remarkably publishable. I guarantee you I can get those out. And so basically, it's natural and important. You need to think about Hill's criteria. If you don't know what Hill's criteria is, write it down and go Google it. But it's the criteria for causality. The weakest among his criteria is this medical mechanism. There's a reason for this to be happening. Once I see the data, I can always come up with a reason why it's happening. You need to limit your analyses and keep track of what you've done. In other words, a priori, I need to say I'm going to do subgroup analysis for age, race, and sex. I need to publish that, and I need to keep it out there. Okay. I need to appropriately report results. By the way, there's nothing wrong with doing the data snooping as long as what you do in your paper is say, by the way, I was data snooping and I found this out. Somebody else please confirm this. Right. When you're a consumer of subgroup analyses, watch out. The most logical and clinically important subgroup may well be one of those that's, one of the 10 that are significant, and that's all they presented. Look to see if your authors said what they were going to do a priori. It's critically important, This is an example, and I'll stop after this for a break, but this is looking at effect modification in ASCOT. And so what you have here is, this is called a forest plot, very common in clinical trials. And so this is the effect for diabetics and non-diabetics, for current smokers and non-current smokers. So when you look at a forest plot, what you want to do is look down here and see how homogeneous the effect is in all the different subgroups. So, wow, look at this. It's not effective in women. How do I interpret that? It looks like the drug, this is a lipid-lowering drug, looks like it doesn't work in women. Well, to begin with, women have a large standard error. And second of all, I'm looking at a lot of subgroups, and I ought to anticipate that one or two of them are going to do screwy things by chance alone. So the authors of this very appropriately said, don't worry about that. They said, should we not treat women? Is it less effective in diabetics? I looked at 18 a priori strata. I found no significant interaction. And to me, this is a statement of the homogeneity of the effect, not that women and diabetics shouldn't be treated. I know the guy who did this study real well, and he got a letter saying that he would never treat any more of his women with this drug on the basis of this, which of course is stupid. Looks like it's time for a break. Anybody? Oh, yes, sir. Yes, ma'am. But sir, ma'am first. Sir, go ahead.
I was just going to ask about multiple hypothesis correction. So what if, say for that female one, if you did a correction for multiple hypotheses and it was still significantly different, would you maybe take that more seriously?
Great question, by the way. So one of the ways, so what you have to do is to protect yourself. Okay, and there are lots of different ways to protect yourself. If what I know I'm going to be doing is looking at 3 subgroups, the most common way to protect yourself is called a Bonferroni adjustment. So instead of saying that it's going to be 0.05 for something to be significant, I'm going to say it's 0.05 divided by 3, so 0.6167. So basically all you're doing is saying, I have this coin that's 5% chance of heads. Oops, it's the wrong coin. I need to get a coin that's 1.6% chance of heads. If I flip that coin 3 times, now I'm back to my 5% chance. Okay. I'm not a big fan of Bonferroni adjustment. It turns out it's incredibly overly conservative, phenomenally overly conservative. I think it's better, my approach is to use good judgment. I'll try to use good judgment. And so how many things did you test? And really, what is the p-value anyway? I mean, I'm not enamored with 0.05. Something is, one of the interesting questions in trials is, if I do a study and it's significant, p is equal to 0.04, and you completely replicate my study and you get a 0.06, have you refuted me or have you confirmed me? And I will argue that you confirmed me. That the 0.05 is just a stupid number anyway. And so basically, I think part of this is to just be aware of how many tests are being done, be aware of how liberal a person is in their approach, and be careful.
In that last example you showed, you said they did 18 subgroup analysis. In those kind of multiple comparisons, does it matter if it's a priori or post hoc? Because you bound to find something if you're making that many comparisons.
No, the reason why a priori makes a difference is a lot of times you see things in analysis that are a surprise. And once you've seen it, it is then inappropriate to come back and attach a p-value to it. In other words, let's say that a good example is, let's say that I'm doing analysis and I notice in my analysis, the way that statistical testing works is I have an hypothesis, I collect data, and I do my test. Okay, if what you're doing is being data-driven, you're saying I have the data, I'm going to get my hypothesis, I'm going to do a test. Once you see that a difference is there, you can't attach a statistical hypothesis, I mean, a p-value to it. It's like if you're riding down the road and you look up in front of you and you say, my God, look at that, drop that license plate in front of me. It is KL8643. Can you imagine how rare it would be to see a license plate which is KL8643? The odds of that are 1 in 10 million. Well, not once I see the license plate, it's not. There's only if I hadn't seen the license plate before. In other words, that's having the hypothesis before I do the test. And so the answer to your question is, it doesn't matter whether it's a priori or not. But within the not a priori, there's the data dredging, which is a horrible thing to do. In other words, I'm just going to look at the data and find things that I'm going to test. The trouble is the people who do that generally don't tell the truth. You know, and so if I'm reading a paper and they say a priori we were going to look at 3 subgroups, age, race, and sex, not only do I know what to adjust for that, I also know this is not somebody who happened to see this strange effect and is reporting a p-value for something they observed post hoc. And so to me, the real power of the a priori is a statement that this person is not cheating. But if you do 3 tests a priori and you do 3 tests post hoc, it's the same mathematics of a significant finding.
So kind of going back to your example, so if you have an RCT that has, you know, the placebo and the treatment arm, and if you have a different investigator who has a different method who is not privileged to any of the data, and they wanted to look at specifically the placebo arm to look for their outcome, and they have a hypothesis coming in, is that, and just want to use it as a sort of observational data, is that considered a priori at that point since they were not privileged to any of the other information and all the specifics?
No, you really are bringing forth a more challenging question, which is the whole field of observational epidemiology.
Okay.
I think of epidemiology as being more goal-driven than hypothesis-driven. And so the answer to your question is, if he hadn't looked at his data and he had this a priori, I think he's absolutely fine.
Okay.
But let's say we have 20 people have different, you know, one person wants to look at statins, one person wants to look at blood pressure, one person wants, you know, all 20 of them come to the same guy and say, give us your placebo data. You know, one of those is going to be significant by chance alone. Break time. Thank you. Is this okay? Too old. I'm 2 hours long. Well, not only that, I mean, don't do statistics first.
You know, it's interesting when they for their course, ask people what they like, what they don't like. People who've had a lot of statistics say don't do it. We've done a foundation. But people who haven't had a lot of statistics say, I always wish they had a little more. And, so that's
Yeah. It's interesting. We've got young ladies who are
my bad.
2-week teaching courses.
I'm not.
The statistics always are on the regular level.
Okay.
Definitely hidden stuff.
Did you go?
No. So my question that I didn't state very well, it seems like a drug company that's trying to gather data for whether to go for it, so they they're competing goals. They want to know whether their drug is efficacious enough that they will be able to find a niche. But they also don't want to miss a big safety problem in the early development.
I mean, you go after adverse and sometimes you find this sort of
whether they want the most homogeneous starting group so that they can see efficacy more clearly and they want more real life.
I think people start off more homogeneous. I mean, but I mean, Mr. Pitt was all about.
Yeah.
Yeah.
Yeah, I mean,
Great intentions. And then they call it Mr. Bill, which is a multiple risk factor intervention trial.
No, that's interesting. I didn't know that. Yeah, that's good.
Yeah, that's cool. That's the best way to replace the whole thing. I was here, so like this, but I could go. Rachel found out how to merge a bunch of. So I think it's awesome. But she's you. Right.
Call you, Shane. Call you, Shane.
I was like, oh, stop this. Say where they go. Yeah. And then,
Hey, you, George Howard, how are you doing? Good morning, how are you doing? Hey, listen, I asked you the wrong question when I said how many people can at Amendment 5. The right question should have been how many people can be on Amendment 4. What you make here attendance.
yeah, exactly. That's what I think.
If you get these calls to you in gene scans, yeah. Gene scans are really get them. That's what it is.
Okay. Okay. Is there kind of payment for those or anything?
If you want to use them, if you want to use them, I'll still make it.
Yeah.
I want to make sure that.
Yeah.
Right.
Fix my case for
Yeah, but different sites setting approved different kind of drives.
Those are very cheap numbers.
So if I give one.
I guess on that spreadsheet, what is the line that you can start with? You just say what's good.
What's the end of them? Eventually, the special coming into a new one.
In other words, you propose at the beginning of it.
So this is over 10,300, whatever. Yeah.
Yeah, but what I want to calculate, so I like a monthly.
So they have released.
And if I do that, then the sites that are putting in the systematic so what I want to do is have everybody have the same. So instead of doing it the first time, everybody Okay, I've got to finish the lecture. Let me finish it and I'll give you a call back. Okay, don't worry about it, man. Everything's cool.
Okay, so, you should, I'm going to reach out and pretty much on the budget.
I'll see what I can do. Let's see if we can get going again, I guess.
Yeah. Ready to go.
I've got about.
I'm just start
Of the zero that I use also the recording time.
So I'm beginning to panic over my outline and where we are, so I'm going to try to speed up a little bit.
have some discussion of data safety monitoring work later, so.
Okay. Anyway, so efficacy and safety, boy, I wish I had more time to talk about this. This one isn't Trials are designed to assess differences in a primary outcome, so it is powered for the efficacy outcome. But let's say that what you have is something that improves an outcome, modified ranking, and therefore it's efficacious, but it kills 20% of the people. Is that an attractive drug? And so this is the efficacy outcome. That's the safety outcome. Would you recommend the treatment? And generally, unsafe treatments are not employed, even if they're quite efficacious. And the teaching example of that, again, is Vioxx. Drug works great other than causing heart attacks. And so the trouble is that the trial is designed to focus on efficacy. And safety is so important. Why isn't it one of the goals? Why don't we have a trial and design it to look at both safety and efficacy and be powered to do both well? And the issue is that, and again, trying to speed up just a little bit, the safety outcomes are more rare than the efficacy outcomes, so the study would have to be ginormous to do both of them. And so therefore it tends to go for the efficacy outcome. And so basically what we do is to have clearly defined efficacy goal, but only gut checks for safety. And we'll talk some about this if we get to the Data Safety Monitoring Board again, if we have time. But in CREST, the first 9 deaths in this study were all in one treatment arm. And if you want to talk about tense periods, it ended up being completely equal at the end of the day. But the first 9 deaths were all in one group. Really hard. Moving on to masking. By the way, it's also called blinding or masking. The idea of masking has come out of the Eye Institute. They sort of object to the term blinding clinical trials. They prefer the term masking clinical trials. I'm serious. This is where it came from. The basic idea is that it needs to be at equipoise, and you want to avoid any preconceived notions that you have a differential benefit. And so basically, people, both the investigators and the patients, assume that the active treatment is better than placebo. This has always been one of my favorite cartoons. Is a member of the placebo group, but physicians want to help people, and so basically, if the evaluator knows that which group is being treated, they'll tend to perhaps look more diligently for events or ignore events in one group versus the other. Likewise, if the patient knows about it, they might be more likely to report events in one group versus the other. And so, equal evaluate. I'm sorry. And so basically, let me go back to that. So basically, the whole idea of this is to keep it pure. Very frequently, you cannot do blinding or masking. The endarterectomy studies are a good example of that. It's hard to, do sham operations on people's necks or something to open them up and, not do anything. Would be ideal from a design standpoint. It'd be hard to get people in the study. When we did PEPI, the postmenopausal estrogen progestin intervention trial, one of the randomized groups was an estrogen-only arm, which you would think women would figure out their own estrogen only. And it turns out at the end of the study, when we asked women what treatment they were on, the identification of their treatment was no better for the estrogen-only arm than the estrogen and progestin arms, which I find amazing. But, it can work sometimes. So basically, you want to avoid bias from the study design. Equal evaluations, one of the important concepts is you want both groups treated the same way as much as possible. If you have one group where people are coming back into the clinic more frequently, you're going to tend to find more events, just simply because, not because more events could be happening, but just simply because they're coming back into the clinic. So basically, if you're doing a study of calorie restriction and it requires weekly classes for training and the usual treatment does not, surgical follow-up, you might want the surgical group, might want the patient to come back in and make sure the wound is healing okay. In all of these cases, it's really important to think through and have both groups treated the same way. If the surgical group is coming back in for an evaluation, bring the medical group back in. If you're doing caloric restriction and you're having classes on food preparation or such, have classes in the other group for safe driving or something. Make something up so that you're getting people handled the same way. Masking to protect randomization. Biases can be avoided if neither the patient or the physician knows about the treatment. It implies that the follow-up schedule is identical. The old term is blinding, the new term is masking. Double mask is, of course, neither the patient nor the investigator. Single mask is usually the patient does not know that the physician does. Again, a term that's not well defined sometimes. A lot of challenges to masking. It's always not positive. Surgical wounds, responses to treatment of, cycling to HRT replacement. A lot of times you'll have lab tests done that can unmask a study. You need to think through that. If that's happening, you need to think through who has access to those labs. One of the things we do a lot, very frequently, is when you have MRs done, we'll have somebody in the site who's not part of the study review the MRs for the patient to make sure they're okay clinically. But that person, in theory at least, doesn't speak to the people who are part of the assessing the outcomes for the trial. If you're doing MRs, you might see a stroke, a silent stroke, and then you might respond to that. And so we wanted to avoid finding them that way. Participation in the treatment, in other words, low-fat diets, exercise, part of that. So one very common approach, if you can't mask, is to do what's called a probe study, a prospective randomized observer blinded study. So basically, if you can't blind the physician or the patient, have the outcome recorded and have a blind person evaluate the outcome. And a good example of that is Kennedy Lee's has a whole center for reading, for doing modified Rankins. And so basically a nurse will come in and do a clinical assessment on the person, and it will be videotaped. And then the videotape is mailed to a place and they review it to assess whether or not what the Rankin score is, without the person who's scoring it knowing what the treatment is, although the nurse knew, you can argue. You can also move towards totally objective outcomes. If your outcome is death, you don't really need to blind the study. I can adjudicate death. It's the more subjective the outcome is, the more important blinding and masking is. As you move towards objective outcomes, this becomes less important. And again, have an independent observer who doesn't know the outcome. Moving into analytic issues, one of the interesting ones, has everybody heard intention to treat? Is anybody who doesn't? I won't do that. So the first time you hear about intention to treat, it's a strange concept. Okay? If everyone here has heard it, but let's assume that we have 100 patients randomized to two groups, surgery and stent. Of those in the surgery group, 10 patients announced the only reason they were in the trial was to get a stent. This actually happens to us all the time. And they demand to have the stent. So instead of getting the surgery, they get the stent. And the question is, do we compare those getting the stent, the 110 getting the stent, to those not getting the stent, or those assigned to the stent versus those assigned to surgery? The first time that you're dealing with the surgeon and you say, if this person dies without the surgery, we're going to count it against the surgery. That does not go down well the first time a surgeon hears that. But the right answer here is the intention to treat, which is, on the surface, it makes perfect sense to analyze those who received the treatment. These are patients where treatment can make a difference. However, the chance of changing treatment is associated with some other trait. Let's say that in my example of people who wanted the stent, that young people who were more educated were the people who were aware of the opportunity for stenting. And so the only people, those 10 people who changed groups, were all high-income young people. And so what you've done is made one group older and dumber than the other group by these crossovers. Okay, and the whole idea of randomization is these two groups are the same. And so to protect randomization, you always, at the end of the study, you always compare those that are that are assigned to the treatment. It is the intention to deliver the treatment. One of the other very interesting and I think very important concepts is one of the reasons the treatment might not work is people might not do it. And so basically, if you have a really great treatment, but everybody declines to be treated that way, the right answer is the treatment doesn't work, no matter what it does in the people that actually receive it. And so not only is it good to protect randomization, but actually the question you're asking is a more important question anyway, which is, if I try to treat somebody this way, is it a benefit to them? Very quickly, statistical power. Wow, I wish I had more time for this one too. So basically, the whole idea of a trial is that there's the truth. And what I do in a trial is I always assume, well, in a superiority trial anyway, that the two groups are the same. Okay? I'm going to do my study, and I'm either going to conclude there's no evidence of a difference, or I'm going to conclude there's a difference. About 90% of our academic lives lives in this column of the null hypothesis. And so basically, if there really is no difference and I fail to reject, I win. I got the right answer. Okay? There was no difference, and I said I can't see a difference. Okay? However, if there is no difference and I can conclude that there is a difference, I've made an incorrect decision. Okay? We're going to call that a type 1 error. Okay? A type 1 error is when I say there's a difference and one doesn't really exist, and I can control that and the design of the study. And it's also called alpha, and alpha is traditionally 0.05. Okay? Which means that 5% of the trials you read are full of hooey anyway because they're spurious findings. Okay? Turns out there's another column, which is the alternative hypothesis is really being true. The drug really works. The drug really works. Okay, so if it really works and I fail to see a difference, I've made an incorrect decision. Okay, the drug really worked and I said it didn't, or I don't see a difference. And that's called a type 2 error. Okay. By the way, that's a very expensive thing to do if you think about in major trials, if you make a type 2 error, an efficacious drug that would be of great benefit will be killed, and it will never be used for the betterment of mankind. So this is a serious mistake. On the other hand, if there really is a difference and I see it, I've got the right answer. I win. Okay? And that is 1 minus beta. I'm either going to say there's a difference or not, and that is the statistical power. So I want to take a minute and make sure you're comfortable with the concept of statistical power, which is, if there is a difference, what's the chance that I say there's a difference? That's the statistical power. Okay, so type 1 error, saying that the null hypothesis is false when it's really true. You need to decide how much risk you want to take. That's alpha. Normally, you don't decide because the journal said that you want to take a 5% chance, and so you're going to get your paper published and take a 5% chance. Okay, type 2 error is failing to say that the null hypothesis is false when in fact it is false. Again, you want to decide how much risk you want to take. That's beta. Okay, normally, just like there's an arbitrary 5% chance for alpha, people who, I don't know where this one came from, is either 0.1 or 0.2 are the numbers for the type 2 error, which is either 80% power or 90% power. Again, totally arbitrary, completely arbitrary, out of the blue. You know, God knows where it came from. So if the null hypothesis is false, you want to say it's false, and beta is the chance of making this mistake, so 1 minus beta must be the chance of making a correct decision, and 1 minus beta is the statistical power. That is the statistical power. Okay? When you're designing a study, there are 5 interrelated numbers. 5 interrelated numbers. The first is, what is the level of significance that I need to say that things are different. Okay, that's called alpha. Okay. The second is the likelihood of seeing a significant difference if one really exists. Okay, that's my beta. Well, really 1 minus beta, the power. The third is the true difference between the groups. This ain't rocket science. Things that are really different are easier to see than things that are close together. Okay? Bigger differences are easy to see. Okay, I'm going to call that delta. Okay. The next is the sample size. Okay, I'm going to call that cleverly N. Okay, and finally is noise that's in the system, and I'm going to call that sigma. Okay, it turns out if these 5 things are related in a function, I'm going to show you what it is in a second. But if you give me 4 of them, I can tell you what the 5th one has to be. Okay, and so basically, if I know any 4, I can solve for the 5th. So if I don't know this, if I know alpha, sigma, N, and delta, then basically I can perform a power calculation to find out what beta would be. Okay? If I don't know delta, but I know the power, alpha, sigma, and N, then I can perform how big does the difference have to be for me to see that it exists? I do a detectable difference calculation. Okay? If I don't know N, but I know delta, power, sigma, and alpha, then I can do a sample size calculation. Okay? So basically, all of these is the same formula. It's just which one do you not know that you're solving for. Okay? And I won't go into it, but this is just the formula. If you're interested in it, ask me later, and I'll show you what it is. I thought I had a slide on it, but I'm going on to something else. Yes.
So sometimes in our clinical trials for cancer drugs, we do 2:1 randomization. It's not always like 1:1.
Mm-hmm.
So it's 2 patients go on the drug and 1 on placebo.
Right.
So is there an adjustment you make for the N based on that?
No, it turns out that the smallest sample size will always be equal randomization. 1:1 will always give you a smaller sample size. The reason for doing 2:1 is largely because people believe the active treatment is good for you. And so if you're walking up to somebody and you say, would you be in my study, there's a 50% chance that you'll get this drug that might help you. You might say no. If I walk up and say, would you be in my study, you have twice the chance of getting the good drug than the bad drug, and you might say yes. So that's the first reason, is to increase participation. The second reason is, if I am interested in the safety of the drug, then what I'd like to do is maximize the number of people who receive the drug. So safety is really an important concept in the trial, which I don't know cancer trials at all. But if safety is really one of the driving forces, then doing a 2:1 randomization gives me more people on the drug and more adverse events, so I can describe the safety profile better. So those are the 2 reasons. Make sense?
Yep.
Okay, so suppose you're halfway through the trial and there's a clear benefit in one of the treatments. Then I'll argue you're both ethically and financially bound to stop the study. Okay? And there are 2 ways, and so basically the deal here is that you need to think about that a priori. If you plot out the p-value, if you came in every morning and plotted out the p-value for the difference between the 2 trials, it would go up and down day by day. And it's a fun thing to do if you're a statistician, actually, because sometimes it gets significant for a while, and then it'll come back down and be non-significant. And eventually the oscillation in the p-value, as the sample size gets bigger, gets smaller and smaller, and you begin to believe it. So basically, if you are doing this interim analysis, it's very important to say, I'm going to look at this time a priori. I'm going to tell you on this date, with this number of patients, I'm going to do a test. Because if I come in and do it every time, if I did it just during one of the times that happened to be significant, and I stop my study, I get a wrong answer. So basically, there are 2 ways to think about reducing the number of patients in a study. The first is called group sequential trials. Okay, this is traditional interim analysis. Okay, the idea here is I plan a big study, and I see if I can stop it early. Okay. Okay, so basically what I'm going to do is I'm going to start my study, and when half the patients are in there, I'm going to do an analysis, and if it's significant, I'm going to stop my study with only half of the people in the study. Plan a big study, see if I can stop it early. Okay? You have to pay a price for that peak. Okay? It costs, the more you look, the more likely you would be to find a difference. And so people have come up, O'Brien, Fleming, Landametz, Pocock, and Pito have all proposed approaches for you to pay for the penalty of the peak. Okay? I love the name of all of these. It's called an alpha spending function. I love that term. And so basically I have 0.05 of alpha here in my hand. And if I want to look early, I'm going to have to take part of my 0.05 and spend it to look early. And now I'm going to have a little bit less alpha at the end of the study. Okay? By the way, anytime in life that people say, these are the 4 correct ways to do this, that is equivalent to saying there is no correct way to do this. Okay? If there was a best way to do this, there would only be 1 rule up there, not 4 rules up there. Okay? The most common of them is O'Brien Fleming. What O'Brien Fleming does is say, if you think about it, how do you want to spend your alpha? Well, one way I could do it is do equal allocations. I could do 0.1, 0.1, 0.1, 0.1, 5 times. That would give 0.01, I meant to say. That would give me a good chance of stopping early. I like that. Okay, probably this is called the Pito rule. Okay? But it also means at the last look, I only have 0.01 of my alpha left, which means at the end of the day, if I have a p-value of 0.02, I would be in the awkward situation of saying not significant, p equals 0.02, which many people would be confused by. So the O'Brien Fleming spending rule says I'm going to just spend the smallest amount and save as much of the alpha as possible for the end of the study. So basically what happens with the O'Brien Fleming is for it to be stopped early, it has to be significant. I'm making this up at 0.001, but I've only spent 0.001, which means I have 0.0499 left at the end of the study. So I'd be unlikely to be caught in that awkward situation of saying it's less than 0.05 and not significant. Again, 4 rules means that there's not a good rule. Each one of them is how you spend this alpha to keep overall 0.05. It's widely accepted, completely valid, but you have to plan your peaks. You have to plan your peaks. The second way, which is one of the hot new trendy areas, adaptive clinical trials, the goal here is to start with a small study and decide how big my study needs to be. Okay? Group sequential is I start with a big study and try to stop it early. Adaptive is I start with a small study and see how much bigger it has to be. Okay, rapidly evolving area in statistical research, very ill-defined term. And people will say, I'm doing an adaptive clinical trial. Trust me, you have no idea what they're doing. I mean, it is so vague, it is unbelievably poorly defined. But they fall into 2 broad domains. Okay, the first domain is adjustment for nuisance parameters. God, what is a nuisance parameter? So I have to guess what the standard deviation of the measurement is. Okay, let's say I'm looking at something that has a continuous outcome. I have to say, and the standard deviation is 10. Well, if I'm wrong about that standard deviation, then I've got a wrong sample size. That was the sigma. So an adaptive trial could estimate that nuisance parameter. Alternatively, I think 5% of the people are going to have events. Let's say I get the study started and 10% of the people have events. My God, my study can be half the size I thought it could be. I'm not talking about what the second group is looking at interim assessment of the differences between the groups. So you're either estimating nuisance parameters or you're estimating treatment effects. Those are the 2 kinds of adaptive trials. To me, the ones that are looking at treatment differences are a bunch of hooey, and you really need to watch out. I mean, even the question that's being asked philosophically, philosophically what you're saying is I'm going to do a trial. I think an important difference is 5, on the Rankin scale between these 2 treatment groups. Halfway through, I've changed my mind. It's really 0.2 is the important difference. And so the idea of changing the rules, I don't like trials that are do adaptation on the basis of differences in treatment. Estimation of nuisance parameters, very well accepted. Jury is still out on the second one. There's a great FDA paper describing the approaches. If you want it, I'll be glad to get it for you. So which approach is better? Is it better to start a large study that you can make it smaller, or a small study and make it bigger? Both of them seem reasonable. You know, which apprise the smaller sample size? It turns out that generally group sequential trials will have a smaller average sample size. In our world, they're called more efficient, which is logistically, politically easier to implement. I assure you, if you're being funded by NIH and you say, I want to do a randomized clinical trial, I don't know how many people are in it. Let me get started, and I'll tell you later how big the study will be. NIH has trouble with adaptive clinical trials. They're still working through how to do that, but that doesn't work well with them to say, I don't know what the budget will be. Trust me, I'll tell you later. Okay? Then other criteria. So which one is better? I'm going to fly through this. This one's easier. You know, basically there's a paper, this is a quote, any adaptive trial, no matter how it's obtained, can be improved upon by a likelihood ratio test that utilizes the same acceptance and rejection region. Basically what that says is group sequential is always better, period. You know, which is politically better? It depends on if you're a farmer, then it might be good to do adaptive trials. Really, that makes sense if you're a farmer. If you're an NIH researcher, as I said, it doesn't make any sense. NIH has trouble with it. I'm flying through this right now. One exception, by the way, Chris Coffey has a great idea. When you're doing these major trials and they're funded by NIH, they never finish on time. The recruitment, you know, my old mentor said there are only 3 problems with randomized clinical trials: recruitment, accrual, and getting people in your study. He says that's the only problems you have. Okay, so you plan this study, you ask the clinics how many patients they have. They say, oh, I have 20 patients a year. You start the study, they have 2 patients a year. Okay, so you get funded for a 5-year grant. You're going to be doing, going back in 5 years or 4 years for continuation to complete your study. Okay, Chris Coffey had what I think is a great idea, is that's when you ought to do the adaptive trial and say, now that we're here and you're getting ready to refund us, do you really want to do it? You know, this is how big our study should have been, kind of thing. So it's really a good idea. And other issues, I'm going to skip over this because I want to get to those. Okay, so basically, I'm a big fan of group sequential, by the way. Adaptive trials, I think, is a very trendy area. The criteria for making adaptive trial decisions is to reduce the sample size of power. Group sequential trials are uniformly superior. Logistical reasons for implementation, group sequential trials seem to have fewer issues. And other issues, it's not clear, by the way, the idea of an adaptive trial, you can think of it as trying to save the study. And maintaining blinding might be important. Let's say you're doing an adaptive trial and you get to the point of adaptation, you say, just kidding, we need 50% more patients. I mean, think of what you just told all of the investigators about, you know, treatment differences and such. And other than sounding like you're really a cool and smart person, which I think saying I'm doing an adaptive trial makes people think you're really smart, I'm not a big fan. Hot new approaches. One of the hot areas is Bayesian analysis. Again, very trendy, very popular. Okay, from a 30,000-foot view, you know, an inappropriately raging debate involves that. Frequentists, which is what I am, I'm a frequentist, think that there's a true difference between the 2 groups. It exists in nature. The difference between people treated medically and people treated surgically is a 5% difference. It exists in nature, God knows it, and it's my job to try to guess it. Okay? Our goal is to guess or estimate what the difference is. Bayesians think that the difference is really a distribution. It's not a number, but a distribution of numbers. that are between the two treatments. And, and by the way, we already know a lot about what difference would be there. And if you think about mounting one of these big trials, it doesn't come out of nowhere. So I've got all these other studies, all this phase 1 and phase 2 studies that are being done. Shouldn't I use that information from those phase 1 and phase 2 studies to better inform my phase 3 study? I could reduce my sample size. So then if you believe that, if I can bring my phase 1 and phase 2 data in and just update it, then basically what I would do is have a smaller phase 3 study, which is really a cool idea. I don't need to say there's substantial prior information to powerfully inform a randomized clinical trial. You don't happen to do one of these on Tuesday because you want to do one on Tuesday. You've got tons of animal and human data that has led up to these phase 3 studies. It's a huge win if you can use it. However, there's not prior information. However, I'm sorry, let me say that again. However, if there's not prior information, there's not a lot of gain from doing a Bayesian analysis. You'll get the same sample size if you do Bayesian and frequentist, unless there's a lot of prior information. Some argue that Bayesian analysis allows adaptive trials to be better implemented. This is true, but adaptive trials can be also implemented under a frequentist standpoint, so that's not a gain. Okay, some argue that Bayesian analysis answers the logical question. That is, I have this information, I want to update it, and it can be easily argued from both approaches that both frequentist and Bayesian give some answers logical questions. Okay, and so the true gain is, again, that opportunity to capture the prior information and reduce the sample size. That's why you would go Bayesian. That's why you would go Bayesian. But is that something we really want to do? We really want to do Bayesian trials. Obviously, I'm biased here. But anyway, I 100% agree Bayesian approaches have the advantage of optimally using the information from the previous trials. It allows a smooth transition from phase 2 to phase 3 trials. That seems like it's a good idea. What kind of fool would not want to use all the information that's not available? And that fool is me, okay? I think that Bayesian phase 3 trials are bad, real bad, okay? And they're bad, real bad for 3 reasons. They're problems about what we know. Okay, let me see. And we all have to agree on what we know. If we're going to inform the phase 3 trial about what we know, we have to agree on what we know that feeds into the mathematical assessment. As clinicians and as scientists, we do a lot of things well. Agreeing is not one of the things we do really well. We tend not to agree on this is important, this isn't important. We have to know exactly the right amount. Let's say we know too much. Well, first of all, if we don't know anything, there's no gain. But if we know too much, I would challenge you to think about the informed consent where you walk in and say, I've got this new treatment. I'm 90% sure the new treatment is better, but I'm not completely sure. Can I randomize you to get the new treatment or the old treatment? And it makes, that's going to be a hard, if you know too much, you'll get a big gain, but you can't do the study. And then this is the most important one, is we might systematically know the wrong stuff. And this is the reason why Bayesian phase 3 trials should not be done. This is the main reason. So I have 10,000 compounds that are going to be moving forward. They go into animal studies, and 5,000 of them get thrown away. They go into baboons, another 2,000 of them get thrown away. They go into phase 1, phase 2, phase 3 trials. I'm now down to 5 compounds that are really going to go into a phase 3 trial. I started with 10,000. The only ones that are going to go into phase 3 are the ones that look very promising. Okay, you don't do a phase 3 trial unless it looks very promising. There are 2 reasons why things could look promising. The first reason is it's promising. And the second reason is, by chance alone, the studies that have led me to this place will have overestimated the efficacy of this study. And so the the treatments that make it to phase 3, by definition, all of their pilot work is biased, every bit of it. Conditional on the fact I'm doing a phase 3 trial, I have bad information in the prior information, because I will systematically take things that are working. One of the 2 reasons they could be working is I have a biased estimate and think it's working when it's not. If I feed in the biased estimate from my priors into my study, I get a biased outcome. And so I think this is really a bad thing that we need to strongly avoid. Are people who disagree with me. And then the final point is, let's stop and think, do we really want to use that information anyway? If I'm saying that this is the definitive trial, this is this is it, this is the definitive trial, I personally think it's a very attractive trait for it to stand on its own. In other words, I don't think it's a good thing to use the information. I think it's a bad, what I want to do is say, I wiped the board clean, and this is the answer I got. And that's the frequentist approach. So I'm going to skip this because I'm out of time. But this is exactly what I was going through. You know, if it's 60-40, I might be able to randomize. If it's 70-30. And again, we pretend like it's the only study, but it's not. And I'm sorry that I'm having to do this. I apologize. And then the very last thing I'll talk about is this. Okay, I don't want to be rude. Is the whole idea of missing data in randomized clinical trials. So again, huge topic, huge topic. The best way to address missing data is don't have it. I mean, that handles it. Really work not to have missing data. It's sort of the statistician's full employment act. But frequently, you know, it's more common than we wish. People don't drop out at random. You know, a great reason for somebody not to come back for the clinic visit is they had a heart attack. Okay? If they don't come back, you might not know they had a heart attack, and the heart attack is the outcome. Okay? And so basically, they don't drop out at random. And assuming that they do give some wrong answers, this is a remarkably hot area. The 2 big approaches to handle it are, you read about the mixed models of Laird and Ware, which is trying to make assumptions about the correlational structure between the different data. And another very, very hot area is multiple imputation, where basically you make up data, or you're missing data, but you do it in a very clever way that doesn't give you inflation of your power. If you do have to do this, buy a statistician and start buying drinks. Don't do these on your own. I mean, get a pro to do either one of these. And so basically, what I'm going to do is skip all these novel designs for a minute and just hit, I know you're going to talk about data and safety monitoring boards later, but I'm just going to hit it as the last point. You know, it's so hard getting these grants. I worked so hard to get one of these NIH grants funded. I get it funded. It's so great. CREST is funded through 2022. Think of, I mean, gosh almighty, I've got job protection until 2022. I'm unlikely to shut down that study because it goes to paying my salary. So I'm not the unbiased person who you should be making a decision about what should be done. You need to have a group that's independent, whose position does not depend on the study to do this. And so basically I'm in a huge conflict, and in addition, I can't see the data. So generally in all of these, there's an independent board, the data safety monitoring board, who is not in conflict and can see the data. And generally 5 to 7 people, but sometimes 3. They generally are not associated with any of the centers. In CREST, we have 144 clinical centers in the United States. I defy you to name the 145th best center in the United States. So basically anybody who's competent is in the study. And so basically finding people who are totally not at any center turns out to be harder than you think it is. It has content experts, biostatisticians, sometimes patient advocates. It's in my world, it is appointed by NIH and reports to NIH, not to the study. basically, it meets generally about every 6 months and reviews the data in the 6-month interval. It's generally unmasked. A lot of times they ask for A-B blinding. In other words, don't tell me what the treatments are. Just put them in 2 groups called one of them A, one of them B. And that way I won't have my biases for which one I think is working. At 6 months, it reviews study progress, data quality, patient safety. Some committees do it differently, but at least when I'm on a data safety monitoring board, I don't want to know what the treatment differences are unless it's at one of the times appointed for the group sequential trial to assess the differences. In other words, don't tell me what the differences are every time we meet. If we only have one interim analysis, only tell me the difference there at that time. I don't want to be put in a position where a difference might exist and I have to say, that's okay, keep on going with the study. I'd just rather not know. And they can advise early termination of the study. Key safety terms: an adverse event is serious if it is life-threatening, hospitalization, disability, birth defects, permanent impairment. It's unanticipated if it's not discussed in the informed consent and it's not expected in similar patients. And it's associated with the treatment if it's caused by the treatment. And so basically you have different combinations of those 3 things. A trial is under regulation to urgently report serious unanticipated events. If something is both serious and unanticipated, the clock starts running, you have to tell the IRBs, you have to tell everybody, basically, for serious and unanticipated. The decision to stop, hopefully stopping rules are in place for efficacy, so that's a non-issue. We're going to decide that before we get started. Okay. Stopping for safety becomes very complex. A lot of people say, what are your safety stopping rules? I don't like safety stopping rules. I like the rule being that basically, can I sleep at night? Is this something that is of such great concern that I think the study should be stopped? Because if what you do is you say if the serious adverse event rate is 3 times bigger in one group than the other, I'm going to stop. And then you find out it's twice as big, but the adverse events are deaths. And I care more about deaths than I do about other things. And so you can't, it's just simply, can you do a gut check and keep the study going? And I know that was 1,000 topics. It was like all spread out. Sorry it was disconnected. Are there any questions or anything in the last minute I have? Yes, ma'am.
Sample size. Even you start with smaller size, you have to come up with a sample size.
Right. The idea of starting with a small sample size is the adaptive approach. Okay. And what you can do is say, I'm going to start with 500 people and estimate different parameters and then decide how big my study should be. Adaptive trials are very trendy. A lot of people are doing them. I'm not a big fan personally. Mainly because of who funds my work, which is NIH, and I assure you they won't do one.
Yeah, but you depend on the power and the arm on it. There are one or two, and depending on that, you come up with a sample
Right.
size.
So the sample size, if I again had more time, I could talk about it. The formula for sample size is the easy part. The hard part is where do you get the data to plug into the formula? Yeah, what are the standard errors? Okay, if you had good data to plug into the formula, you wouldn't need to do the study because you would know what the difference is, you'd know what the standard deviation is. By definition, if you had good data and you could do a good sample size calculation, the study wouldn't need to be done. Okay, so that means that every time you do a power calculation, it is a swag, scientific wild ass guess. I mean, you're just pulling something out of the air to spend $50 million on, okay? And so basically you end up doing things like talking about how big does the treatment difference have to be considered clinically important. In other words, if the difference between surgery and medicine is 2%, then people will not change their practice. They'll only change their practice if it's 3 or 4%. So I define the delta in that formula as 3 or 4% because that's how big it would have to be to, the standard deviation I don't have. what do? Well, very commonly you guess. You go out and you do sensitivity analyses and say, if it's 10%, which is what I think it is, I need 1,000 people. If it's 12%, I need 1,200 people. You know, am I comfortable that it's not that much bigger? And again, people make a huge deal out of the sample size calculations. Trust me when I say this as somebody who produces them. It is the biggest guess you've ever seen in your life. I mean, it is amazingly sensitive to the assumptions on data that you clearly don't have. Anyway, thank y'all.
Thank you.
Thank you.
Sensitivity analysis, I think of as trying to meet a different end. So sensitivity analysis would be what would happen if things were slightly different. Okay. Subgroup analysis is saying, are men and women, you know, do I have the same effect? The sensitivity analysis, a good example of that might be, let's say that my outcome is the modified line function. And, you know, what would a sensitivity analysis would be, well, what happens if I have a different outcome? Let's say I'm using instead of not the same answer. Secondarily, I think that's analysis. If I would have done things differently, would I have gotten the same answer?
So that's obviously isn't going to
I was just wondering,
It happens in the summer.
Okay.
It happens in the summer. I think Cutter's teaching the introductory to this. Yeah, no, since School of Public Health, you call Della Daniel, and she'll know about it. She's our registrar. Yeah, but you have to ask there.
Okay.
It doesn't involve me, but I'm not.
Okay.
Different people have.
Yeah.
Differently. I think a lot of too, so many people are registered.
Right.
So a lot of people are. It's a good place. Cutter's wonderful.
Okay, so that's part of the biostatistics.
Yeah.
Is it graduate level?
Yeah, it's a graduate level. Yeah, the one semester one is a master's level, and the 2 semesters is a doctoral level, which is, you probably don't want to do that much, have a lot of complications. But one semester is basically what I've done here at this level.